Respected teacher and my dear friends, today I am here to present my seminar topic. My seminar topic is the general properties of microorganisms. The contents are the general properties of bacteria, fungi, algae, protozoa, rickettsia, chlamydia, and their characterization and classification. First of all, what is microorganisms? Microorganisms, also known as microbes, which are microscopic organisms that are too small to see with the human eye. Examples: bacteria, fungi, algae, protozoa, and viruses. Microorganisms can be unicellular, that means single cell, or multicellular, that means cell colony, or acellular, that means they don't have a cell. And they found in water, soil, air, and the human body. They are very essential to life on earth and can be used to grow crops and to prepare bioactive molecules and also for regulating the climate. The study of microorganisms is called microbiology. Most of these microorganisms are helpful, such as bacteria that aid digestion, and scientists believe that a diverse microbial population helps the body to resist the diseases. General properties of microbes. Cell structure. Bacteria are prokaryotic, while fungi, algae, and protozoa are eukaryotic. Prokaryotic cells have a different cellular organization than the eukaryotic cell. Then cell walls. Fungi and algae, except euglenoids. In the classification algae, except euglenoids have rigid cell walls, while protozoa lack a true cell walls. Then shape. Bacteria is commonly seen in spherical, rod shaped or spiral shape, and the size of the bacteria are generally 0.5 to 5 micrometers in width. Then movement. Some bacteria have flagella, which are whip-like structures and that help them to move through liquid. Their reproduction. Some bacteria produce spores, which are reproductive bodies similar to seeds in plants. Nutrition. Fungi are organisms that get nutrition from plants, food, and animals. Cell composition. The cell walls of all true bacteria contain peptidoglycan. Peptidoglycan means it is a major component of gram-positive bacteria. It is made up of polymer of sugar residues, and it is a cross linked by peptide bridges. Okay. Then gram staining. Gram staining means it is used to diagnose the bacterial infections and can also help to identify the fungal infection. Here, this gram staining, some bacteria are gram positive, meaning that they take on a purple color when stained, while others are gram negative. Then the general characteristics of bacteria. Bacteria are microscopic, single-celled prokaryotic organisms whose cells lack a nucleus or nuclear membrane. The prokaryotic means they lack a nucleus or nuclear membrane. And the bacteria may be found in different shapes, and also they have a unique cell walls. They reproduce by binary fission and exist in most environments on earth. Binary fission means it is a mode of asexual reproduction and the cell is split into two identical daughter cells. Here is a picture showing that having different shapes of bacteria. Then bacteria species vary in size. The smallest bacterium, Mycoplasma, are about 0.3 micrometer in diameter and approximately the size of the large viruses, pox viruses. Escherichia coli. That means E. coli. E. coli bacterium of about 1.1 to 1.5 micrometer wide by 2 to 6 micrometer long. Most spherical bacteria have diameters of 0.5 to 2 micrometer and row-shaped cells are generally 0.2 to 2 micrometer wide and 1 to 10 micrometer long. Examples: E. coli, Salmonella typhi, Vibrio cholera, Staphylococcus aureus, Bacillus anthracis, Clostridium tetani, etc. Next, we have to go to cell structure of a bacteria. This is cell structure of a bacteria. The outer layer is known to be capsule, and at most that there is a cell wall, and between the cell wall and plasma membrane there is a periplasmic membrane, then nucleoid where the DNA is contained, and the cytoplasm where all the components are placed, then ribosomes, and then inclusion bodies, that blue color, then the flagella and pilus. Next, we have to move on to the functions of a bacterial cell structures. The cell wall. Cell wall gives bacteria shape and rigidity, and also it protects from the lysis in dilute solution. Lysis meaning rupturing. Rupturing from the dilute solution. It gives protection. Cell wall gives a protection from lysine. Then plasma membrane. Selectively permeable barrier, mechanically boundary of cell, nutrient and waste transport, then location of metabolic process. Respiration is done through the plasma membrane. Then periplasmic space, that is between the cell wall and plasma membrane. This is space known as periplasmic space, which contains hydrolytic enzymes and binding proteins for nutrient processing and uptake. Then nucleoid. The nucleoid is the localization of genetic material, that means DNA, which carry instruction for all cell activities. Then inclusion bodies, it store carbon, phosphate and other substances. Then ribosome, it synthesizes proteins. Then capsule and slime layer, it resists to phagocytosis. Phagocytosis means which kill microorganisms or ingest foreign material and remove dead cell. That is resistance to phagocytosis and adherence to surfaces. It is an immune cell. That means that is phagocytosis. That is an immune cell. Then fimbriae and pilus, it is an attachment to surfaces and bacterial mating. It is used for bacterial mating and also for the attachment to surfaces. Then flagella, it is used for movement. Then endospore. Endospore means it can used for the survival under the harsh environmental condition. Next, we have to move to classification of bacteria. Bacteria are grouped by four main characteristics. Then first one is gram reaction, gram positive and gram negative. Then shape. Then the third one is atmospheric requirements, and fourth one is endospores. The first one, gram reaction. Gram reaction is gram positive and gram negative. Here is a table shows that the difference between gram positive and gram negative. Cell wall in gram positive, single layered with 0.015 to 0.2, and in gram negative it is a triple layered with 0.0075 to 0.012 thick. Then the rigidity of cell wall, it is rigid due to the presence of peptidoglycans, and in gram negative it is elastic due to the presence of lipoprotein polysaccharide mixture. Then chemical composition. Chemical composition means here peptidoglycan, polysaccharide, teichoic acid are present, and while in gram negative there is peptidoglycans is to 3 to 12%, rest is polysaccharides and lipoproteins. Teichoic acid are absent. Then outer membrane. Outer membrane is absent there, and in gram negative it is present. Then periplasmic space, in gram positive bacteria there is absent, and in negative it is present. Susceptibility to penicillin. In gram positive they are highly susceptible. Highly susceptible means the bacteria or fungus causing an infection can't grow off if penicillin is present. That means it is, if penicillin is present it can't grow. That means highly susceptible. While in gram negative they are low susceptible. Then nutritional requirements, relatively complex in gram positive bacteria and relatively simple in gram negative bacteria. Flagella contain two basal body rings, while in gram negative contain 4 basal body rings. Lipid and lipoproteins are low in gram positive and lipid and lipoproteins are high in gram negative. Lipid means a fatty or waxy or oily material that is insoluble in water. Lipoprotein is a complex particles made up of lipids. That is mean by lipid and lipoproteins. Then lipopolysaccharides are absent in gram positive and present in the gram negative bacteria. Here is a figure showing that the difference between gram positive and gram negative bacteria. This is the outer membrane, that purple color in gram positive, and this is a cross-linked structures, that is outer membrane of gram negative bacteria. That is a periplasmic spaces between the cytoplasmic membrane and the outer membrane. This is all the difference between gram positive and gram negative bacteria by figure. Then shapes. The three main shapes of bacteria are cookie, that means spherical shape; bacillus, that means roll shaped; spirilla means spiral or helical shaped; vibrio means coma shaped. For examples of all these are cookie, Streptococcus pneumonia; bacilli, example E. coli; spirilla, Spirillum voltanus; vibrio, Vibrio cholerae. The shape of bacteria affect their ability to move, attach and cause diseases in human and other animals. These are the different shapes of bacteria: spherical, rod-like, spiral-shaped and coma-shaped bacteria. Then atmospheric conditions. Obligate aerobes and anaerobes. The bacteria which are obligate means strict. Obligate aerobes, which can grow only with an abundant supply of oxygen in their environment for growth and survival. That means they need oxygen to grow. Then those are obligate and anaerobes, which are killed by oxygen. Obligate anaerobes means that they will be killed by the presence of oxygen. In fact, oxygen is toxic for them and these organisms cannot tolerate oxygen in their environment. Then facultative anaerobes. Organisms that can survive and grow in the absence as well as in the presence of molecular oxygen. Group of microorganisms can survive and grow in both aerobic and anaerobic conditions. Such organisms are called facultative organisms. Facultative organisms can switch their metabolism between aerobic respiration, anaerobic respiration or fermentation based on the oxygen availability. Then endospores. An endospore is a dormant, tough and non-reproductive structure produced by some bacteria in the phylum Bacillota. The name endospore is suggestive of a spore or seed-like form. Endo means within, but it is not a true spore, that is not an offspring. It is stripped down and dormant form to which the bacterium can reduce itself. Endospore formation is usually triggered by a lack of nutrients and usually occurs in gram-positive bacteria. In endospore formation, the bacterium divides within its cell wall and one side then engulfs the other. When the environment becomes more favorable, the endospore can reactivate itself into a vegetative state. Most types of bacteria cannot change to the endospore form. Examples of bacterial species that can form endospores include Bacillus cereus, Bacillus anthracis, Bacillus thuringiensis, Clostridium botulinum, and Clostridium tetani. Endospore formation is not found among archaea. Here is a figure about the endospore. In environmental conditions, if environmental conditions become unfavorable for the growth, such as they have a lack of food, a bacteria may produce an endospore. A tough shell forms around the cell's nucleus and some of the cytoplasm. Here, this is a figure. It's the outer coat and the inner portion is a cytoplasm where all the components are placed, and there is a hereditary material also, and outer that blue color is a endospore. Next, we have to move to general characteristics of fungi. Fungi are eukaryotic organisms that include microorganisms such as yeast, molds, and mushrooms. These organisms are classified under kingdom fungi. The organisms found in kingdom fungi contain a cell wall and are omnipresent. They are classified as heterotrophs among the living organisms. Heterotrophs means sometimes it may be parasite that it which lives on the base of other living organisms. Appearance of black spots on bread left outside for some days, and the mushrooms and the yeast cells which are commonly used for the production of beer. Are Saccharomyces cerevisiae and bread or yeast. That is also a fungi. Black spots on bread and the mushrooms, the yeast cells are comes under the fungi. They are also found in most skin infection and other fungal diseases. If you observe carefully, all the examples that we cited involve moist conditions. Thus, we can say that fungi usually grow in places which are moist and warm enough to support them. So, they need moist condition. Where the moist condition and warm enough is there is a chance to grow if fungi. This is a structure of fungi. This is the outer cell wall and the bud, perixosome, septum, cytoplasm, Golgi bodies, mitochondrion, ribosome, nucleolus, SCR, lysosome, vacuole, bud scale, etc. Golgi bodies are which produce energy and proteins. That is here present. Then we can move to the structure of fungi. Almost all the fungi have filamentous structure except the yeast cell. They can either be single-celled or multicellular organism. Fungi consist of a long thread-like structures known as hyphae. These hyphae together form a mesh-like structure called mycelium. And fungi possess cell wall which is made up of chitin and polysaccharide. The cell wall comprises of protoplast, which is differentiated into other cell parts such as cell membrane, cytoplasm, cell organelles, and nuclei. The nucleus is dense, clear with chromatin threads. The nucleus is surrounded by a nuclear membrane. Here, hyphae are a filamentous structure that make up of fungi, and septa are the internal walls that divide hyphae into cells. Then the characteristics of fungi. Important characteristics of fungi are eukaryotic, non-vascular, non-motile and heterotrophic organisms. They may be unicellular or filamentous. They reproduce by means of spores. Fungi exhibit the phenomenon of alteration of generation. Fungi lack chlorophyll and hence cannot perform photosynthesis. Fungi store their food in the form of starch. Biosynthesis of chitin occurs in fungi. The nuclei of the fungi are very small. The fungi have no embryonic stage. They develop from the spores. The mode of reproduction is sexual or asexual. Some fungi are parasitic and can infect the host. Fungi produce a chemical called pheromone which leads to sexual reproduction in fungi. Examples include mushrooms, molds, yeast, etc. Here is a figure of mushrooms and examples of fungi. Then the classification. Kingdom fungi are classified based on different modes. Classification on the basis of nutrition. Kingdom fungi can be classified into three groups: saprophytic, parasitic, and symbiotic. Saprophytic means they obtain their nutrition by feeding on dead organic substances. Example, Rhizopus, Penicillium, Aspergillus. Then parasitic. They obtain their nutrition by living on other living organisms. That means plant and animals. And absorb nutrition from their host. For example, Taphrina and Puccinia. Then symbiotic. These fungi live by having an interdependent relationship with other species in which both are mutually benefited. For example, lichens and microrrhizia. Lichens are symbiotic association between algae and fungi. Here both algae and fungi are mutually benefited as fungi provide shelter for algae and in reverse algae synthesis carbohydrate for fungi. This is the best example for symbiotic. Then mycorrhiza is a symbiotic association present between fungi and plants. Fungi improve nutrition uptake by plants whereas plants provide organic molecules like sugar to the fungus. Then based on the spore formation, kingdom fungi can classified into the following bases: zygomycetes, ascomycetes, basidiomycetes, deuteromycetes. Zygomycetes means they are formed by the fusion of two different cells. The sexual spores are known as zygospores, while the asexual spores are known as sporangiospores. The hyphae are without the septa. Example, mucor. Ascomycetes, they are also called sac fungi. They can be coprophilous, decomposers, parasitic or saprophytic. The sexual spores are called ascospores. Asexual reproduction occur by conidiospores, for example, Saccharomyces. Then basidiomycetes. Mushrooms are the most commonly found basidiomycetes and mostly live as parasites. Sexual reproduction occurs by basidiospores. Asexual reproduction occurs by conidia. Budding or fragmentation, etc. Asexual reproduction occurs by conidia and maybe it is budding or fragmentation. For example, agaricus. Then deuteromycetes. They are otherwise called imperfect fungi as they do not follow the regular reproduction cycle as the other fungi. They do not reproduce sexually. Asexual reproduction occurs by conidia. For example, trichoderma. Then the general characterization of algae. Algae include a group of about 13,000 species which are generally aquatic. That means both freshwater and marine. The size varies from microscopic forms to filamentous form. All the members of algae are chlorophyllous, non-vascular and thallophyte. Thallophyte means plant-like organisms. Example for algae. That means they are not truly a plants but a plant-like organism. And thalloid means have plant body but do not differentiate it into stem stones, stem leaves and roots, etc. That means that is thalloid. Okay. Then all the members of algae are chlorophyllous. Then they contain the pigments such as chlorophyll A, carotenes, xanthophylls. Additional pigments occur in specific groups. Mucilage protects algal thallus from dissection and from epiphytic growth. Vascular and mechanical tissues are absent. Then this is the structure of algae. These are the all components of algae. The vegetative and asexual modes of reproduction are very common. Asexual spores are of two types, mitospores and meiospores. Mitospores means they are formed by mitosis and meiospores means they are formed by meiosis. In mitosis, the mitospores are asexual spores that have the same set of chromosomes as the parent body. In meiosis or meiospores, they are produced by meiosis. Meiospores are haploid spores that show genetic variation and to help the species adaptation. They are easily dispersed in aquatic habitat, actively if motile and passively by water currents if non-motile. Their sex organs are not jacketed. Sexual reproduction only occurs towards the end of the growing season. And the embryonic stage absent, which is a characteristic feature of Thallophyta. Sexual reproduction involves isogamy, anisogamy and oogamy in different groups. Different types of life cycle occur: haplontic, haploid phase dominant, diplontic, diploid phase dominant, and haplodiplontic. That means both haploid and diploid phase equally dominant. Haploid means they have 23 chromosome. Diploid means they have 46 chromosome. That is mean by haploid and diploid. Then Classification of algae. Classification is based on the color. Chlorophyceae green algae, also known as green algae, they contain chlorophyll A and B pigments. Include Chlamydomonas, Spirogyra and Cara. Then Phaeophyceae, they are brown algae. These algae are predominantly marine and contain chlorophyll A, C, carotenoids and xanthophyll pigments. Examples include Dictyota, Laminaria, Sargassum. Rhodophyceae, they are red algae. These algae contain chlorophyll A and D and physoceratoin pigments. Examples include Porphyra, Gracilaria, Gelidium. These are the three variety types of algae: green algae, brown algae and red algae. Then next we have to move to general characterization of protozoa. Protozoa are unicellular eukaryotic heterotrophic organisms. They either free living or parasites. There are around 65,000 species of protozoans categorized in different groups and they lack a cell wall. There are many different cell organelles that perform varied tasks performed by different organs in higher animals. Example, mouth, anus, intestinal tract, etc. There are many protozoa that cause various diseases in animals and humans. For example, Plasmodium, that is a malarial parasite, and Trypanosoma, that is a sleeping sickness, and Trichomonasis. These are the different types of protozoa causing various diseases. Then the protozoa have many stages in their life cycle. Some of the stages of this life cycle are infectious. The cyst stage is dormant and resistant to environmental stress. The trophocyte stage is reproductive and causes diseases. Then they found in aquatic environment. They live in freshwater or oceans. Some are living and some are parasitic in plants and animals. Mostly they are aerobic, but some are anaerobic and present in the rumen or human intestine. Some of these species are found in extreme environments like hot springs. Some of them form resting cysts to overcome dry environments also. The size and shape of protozoa are greatly from 1 micrometer to large enough and can be seen by the naked eye. The shell of unicellular foraminifera can have a diameter of 20 cm to 90 cm. Foraminifera is a single-celled organism that having a shell. That is the shell of the unicellular foraminifera having a diameter of 20 cm to 90 cm. They lack a rigid cell wall, so they are flexible and found in various shapes. Cells are enclosed in thin plasma membrane. Some of the species have a hard shell on the outer surface. In some of the protozoan, especially in ciliates, the cell is supported by pellicle. Pellicle means it is a thin skin or a film covering for a certain protozoa. It is just a covering skin, which may be flexible or rigid and give organism that the definite shape and also it help in the locomotion. This is the cell structure of protozoa. They have contractile vacuole. Contractile vacuole is used for osmoregulation. That means to collect and pump excess water out of the cell. That is the use of contractile vacuole. Others are cilia, cytoplasm, anal pore, food vacuole, oral groove, micronucleus and macronucleus. Then the cell structure. They are unicellular having a eukaryotic cell. The metabolic functions are performed by some specialized internal structures. They mostly have one membrane bound nucleus in the cell. The nucleus has diffused appearance due to scattered chromatin. The vesicular nucleus contains a central body called endosome or nuclei. Nuclei of apicomplexans have DNA, where amphibians like DNA in their endosome. Cilia have micronucleus and macronucleus. See, they have micronucleus and macronucleus. There are two types of nucleus. The micro is very small and the macro is little big. The plasma membrane encloses the cytoplasm, and other locomotor processions like flagella, pseudopodia, and cilia. The locomotion is used by the cilia and flagella and also pseudopodia. Some of the genera have membranous envelope called pellicle, which gives a definite shape to the cell. That pellicles means I have already taught it is a thin skin film or a film covering for a covering. It's just a covering. In some protozoa, epibiotic bacteria attach to the pellicle by their fimbriae. Then the cytoplasm is differentiated into outer ectoplasm and the inner endoplasm. Ectoplasm is transparent and endoplasm contains cell organelles. Two type of cytoplasm is there, outer ectoplasm and inner endoplasm. Then some of protozoa have cytosome for ingesting food. Food vacuoles are present where ingested food comes. Then ciliates have a gut, a body cavity which opens outside. The central vacuole is present for osmoregulation that removes excess water membrane bound cell organelles like mitochondria, Golgi bodies, lysosome, and other specialized structure are present. Nutrition. Protozoa are heterotrophic and have holocytic nutrition. They ingest their food by phagocytosis. Some of the protozoan groups have specialized structure called cytosome for phagocytosis. Cytosome is a specialized structure for phagocytosis. Okay. The pseudopodia of the amphibians help in catching the prey. Then thousands of cilia present in ciliates drive the food-laden water into the gut. The ingested food comes to the food vacuole and gets acted on by the lysosomal enzyme. The ingested food gets distributed throughout the cell. Their locomotion. Most of the protozoa species have flagella, cilia or pseudopodia. Sporozoa, which do not have a locomotor structure, have subpellicular microtubules which helps in the slow movement. Then life cycle. The life cycle of most of the protozoa alternates between dormant cyst stage and proliferatory vegetative stage. Example, trophocytes. The cyst stage can survive harsh conditions without water and nutrients. It can remain outside the host for a longer duration and get transmitted. The trophocyte stage is infectious and they feed and multiply during this stage. Reproduction. Mostly they are reproduced by asexual means. They multiply by binary fission, longitudinal fission and transverse fission or budding. In some of the species, the sexual reproduction is present. The sexual reproduction is by conjunction, syngamy or gametocytes formation. Then the classification of protozoa. Protozoa are divided into four major groups based on the structure and part involved in the locomotion. Masitogophora or flagellated protozoans. That means they are parasite or free living. They have flagella for locomotion. Their body is covered by a cuticle or pellicle. Freshwater forms have a contractile vacuole. They are reproduced by binary fission and or longitudinal division. Examples are Trypanosoma, Trichomonas, Giardia, Leishmania, etc. Then Sarcodinia or amoeboids. They live in the freshwater, sea or moist soil. The movement is by pseudopodia. They capture their prey by pseudopodia. There is no definite shape and pellicle is absent here. The contractile vacuole is present in the amoeboids living in freshwater. Reproduction is by binary fission and cyst formation. Examples amoeba, entomoeba, etc. Then sporozoa or sporozoans. They are endoparasitic. They do not have any specialized organ for locomotion. The pellicle is present which has a subpellicular microtubules and that helps in the movement. Reproduction is by sporocyte formation. For example, Plasmodium, Myxodium, Myxedium, Nosemia, Globidium, etc. Coelophora or ciliated protozoan. They are aquatic and move actively with the help of thousands of cilia. They have fixed shape due to the covering of pellicle. They may have tentacles. In the subclass Suctoria, contractile vacuoles are present. Some species have an organ for defense called trichocytes. Trichocytes is used for the defense. They move with the help of cilia, and the movement of cilia helps in taking food inside the gullet. They reproduce by transverse division and also form cysts. Examples: Paramecium, Vorticella, Blandidium, etc. Many of the protozoans are parasite and disease-causing pathogens. Disease caused by protozoa are malaria, Plasmodium falciparum, P. vivax, Plasmodium vivax, and Plasmodium malaria, etc. This is a figure of a Plasmodium. It's a figure of a protozoa. Then the general characterization of Rickettsia. Rickettsia bacteria is a genus of non-motile, gram-negative and non-spore-forming bacteria. It is greatly pleioformic bacteria that may occur in three forms. The three forms are cocci, bacilli and threads. Cocci have a diameter of 0.1 micrometer and bacilli which have a length of 1 to 4 micrometer and threads which are up to about 10 micrometer long. The term rickettsia has nothing to do with rickets, the deficiency disease that occurs due to lack of vitamin D. That means that it had any connection with the rickets, rickettsia and rickets are totally entirely different. Rickets is a deficiency disease that due to the lack of vitamin D. Rickettsia was named after Howard Taylor Ricketts to credit his work on tick-borne spotted fever. Rickettsia commonly applies to any members belonging to the order Rickettsalia. They belong to the phylum Proteobacteria. Proteobacteria, for example, Pseudomonata. Then, the Rickettsia is a small bacteria that approximately range from 0.3 to 0.5 micrometer in their diameter and from 0.8 to 2 micrometer in their length. They are notably smaller than the eukaryotic cell, which roughly range from 10 to 100 micrometer in diameter. The variation in the size of these species can be because of their type or the general shape of the bacteria. Some bacteria can be spherical or coccoid in shape like Rickettsia rickettsii. Some can be rod-shaped like Rickettsia typhi. Several species found in the genus are pleomorphic and hence have the capability of changing their shape and depending on environmental conditions. When they are observed under a microscope, these species can appear singly or in pairs. They may also form strands. Rickettsia is a gram-negative bacteria and hence has the presence of thin peptidoglycan layer in their cell wall. Just like gram-negative bacteria, the cell envelopes contain three main layers. This includes inner cell membrane, the thin cell wall which surrounds the inner membrane and the outermost membrane surrounding the cell wall. Although Rickettsia is said to be gram-negative, the observation may differ when they are stained in the gram stain. Due to this reason, other stains such as Gisma are used often for their identification. It's a figure of Rickettsia. This is a cell structure. They have pores in the outer membrane. Chromatin material DNA is shattered through the cytoplasm. Nucleoplasma, endoplasmic reticulum is shown. The nucleus, the nuclear envelope, outer and inner membrane are also can be seen in this figure. Then the other structural characteristics of Rickettsia bacteria involve the cell wall of the bacteria consists of an acid called diaminopimelic acid. There is no presence of teichoic acid in Rickettsia because they are gram-negative bacteria. Rickettsia species have the presence of lipopolysaccharides, that means LPS, that further consist of lipid and polysaccharide. Genomics. Some parts of the rickettsial genomes resemble the ones of mitochondria. The deciphered genome of the R. Rickettsia provaski species is 1111523 BP in length and it is said to contain 834 genes. It consists of number of genes that are required for anaerobic glycolysis or genes that are involved by the biosynthesis and regulation of amino acids and nucleosides. There has been a recent discovery of another parallel between rickettsia meaning and viruses. That this development may become a basis to fight HIV infection. It has appeared that the human immune response to the scrub typhus pathogen, Orindia sutskemushi, provides an effective that is beneficial against the progress of HIV infection. This negatively influences the virus replication process. Then the classification of rickettsia. The classification of the rickettsia bacteria was made into three groups by method called serology, which is scientific study of body fluids and serum. These three groups were namely spotted fever group, typhus group and scrub typhus group. The grouping was then confirmed by the DNA sequencing method. Human pathogens are included in all three of these groups. The scrub typhus group, the third group, was later reclassified as a related new genus called Orindia, but they still belong to the in the order Rickettales and are therefore grouped with the other rickettsial diseases. Rickettsia are noticed to be more widespread than they are previously known to be. They are said to be associated with arthropods, leeches and protists. Next, we have to go to general characteristics of chlamydia. Chlamydia is a genus of pathogenic gram-negative bacteria that are obligate intercellular parasites. Chlamydia infections are most common bacterial sexually transmitted diseases in humans and are the leading cause of infectious blindness worldwide. They are capable of independent reproduction because they do not synthesize ATP in its development cycle using the host cell metabolic pathways. The life cycle of these microorganisms is original, unique among bacteria and lasts from 24 to 48 hours. Chlamydia antigens consist of four groups: group specific, species specific, type specific and subspecies specific. The group of species specific antigens consist MOP, MOP and heat shock protein. Chlamydia trachomatis is a potent immunogen stimulating the immune processes of microorganisms in the course. See trachomatis infection, that means chlamydia trachomatis infection. The response mechanisms involved are non-specific, specific, humoral and cellular. Chronic infection is characterized by maintenance of microorganisms in the host cell. Inflammation is formed in less time and with increased intensity and has a rapid immune response on the part of previously sensitized lymphocytes. C. trachomatis infection are most common bacterial sexually transmitted infection. It represents an important clinical problem for doctors in many areas of medicine such as dermatology, venerology, ophthalmology, gynecology and obstetrics, rheumatology and others also. Chlamydial infections are important pathogen in medical practice not only because they can cause diseases in various fields of medicine but also because of the large proportion of the population suffering and exposed to the chlamydial infection are characterized by multifocality and polymorphism changes. Chlamydia causes inflammation in the adult urethra and cervixes with the possibility of serious complication and cause perinatal infections in infants. Prenatal infections in infants. This is a structure of chlamydia. This is a microscopic view of chlamydia. Then the classification. Members of the genus Chlamydia are gram-negative bacteria, while the majority of these bacteria have a thin peptidoglycan. The peptidoglycan of these species is not easily detectable. In C. pneumonia and C. trachomatis, however, genome studies have shown that they encode for proteins that may involved in the synthesis of peptidoglycan. In addition, they encode for a major outer membrane protein, a protein located on the surface of C. trachomatis and C. pistachii. This is an important protein used for serological identification of different forms of C. pistachii and tree C. Trachomitis. The absence of peptidoglycan in chlamydia bacteria has been suggested, suggested to be an evolution adaptation that allows them to effectively invade and thrive in the cells of their respective hosts. Infective and reproductive forms of chlamydia include elementary bodies, that means EB, and reticulate bodies. Some of other morphologic characteristics of chlamydia include Chlamydia trachomatis and Chlamydia pneumonia. In Chlamydia trachomatis, they are non-motile, lack structures required for movement, and their shapes vary from cockade to roll-shaped, and the reticulate body ranges from 800 to 900 nanometer in diameter, while the infection bodies ranges from 300 to 400 nanometer in diameter. In Chlamydia pneumonia, they exhibit a cocoon shaped which may be elongated. Their elementary bodies are pear-shaped and range from 200 to 400 nanometer in diameter. So here is the classification of microorganisms, various microorganisms. So my seminar is here is finished. Thank you for giving me this wonderful opportunity and have a nice day.